I'm Kay Tai, the Wiley Vale Chair and HHMI investigator here at the Salk Institute. How do you know if something's good or bad? Let's say something happens, an alarm goes off, and then a few minutes later you discover there was a fire, okay? So how do you assign the relationship between that fire alarm and the fire, or even just the evacuation? How does the brain know to assign credit of one stimulus to a specific outcome? Because we're asking about linking specific valence assignment, whether this is a positive or negative outcome, to a cue that happened seconds ago that cellular basis of memory as we currently understand it just doesn't explain. We've known for a long time that there are different neurons that would represent positive or negative environmental stimuli. We just didn't know how those associations were formed. And when you're learning about a new association and trying to assign valence to a specific cue, that's really what the study was about.
So valence representation of positive and negative value is essentially a critical component of memory formation. It's where we determine and remember whether something is good or bad.
What we already know is that the amygdala is sort of this branching point where information about positive associations, things that are good, are going to be fed down one downstream pathway, and information about things that are bad for us are going to be fed down another. So, okay, it's great. It's almost like you're coming down the railroad tracks, information's coming in, and then you need to be sent one way or the other. The big question is, how are we shifting the tracks to go one way or the other? What is the signal that says, okay, this is good, route it here, this is bad, route it there?
The way we study valence assignment in our laboratory is to use this behavior paradigm called Pavlovian conditioning task. Essentially, we train the mice to associate different auditory tones with either positive and negative stimulus. And we selectively monitored and manipulate the neurotensin signals in the amygdala while the mice were performing this learning task. We found that neurotensin acts as a critical switch to selectively turn on either positive and negative learning.
We do a lot of things with our brains and our minds, but still assessing what is good for us and bad for us is gotta be the number one priority for survival. If there's any perturbation to our ability to determine what's good or bad, this can be manifested as a mental health disorder. Understanding how this happens on a fundamental level is hugely relevant to both our daily lives in health and disease.